Yeah, that works, but the pointer,
The laser pointer.
the laser
you know,
pointer
it's-
is
Hello, everyone. Hello to folks that are here in the room and those that are joining us online. My name's Eric Norberg. I'm the Dean of Libraries here at UT Martin, and this presentation is part of our UT Martin Research Series. As I often say at these events, a lot of people think of UT Martin as a teaching college, and we do a lot of great teaching on this campus. But all of our faculty are expected also to be involved in research activities. And very often that work is kind of overlooked by some folks who don't understand what's going on and what's being done. So we developed this series with the Provost's Office, Vice Chancellor for Academic Affairs, to kind of highlight the different research that's being done by our faculty across campus, and in many cases the research that they draw students into. And I know we have several students today who've worked with Dr. Brumah. So for those that haven't been here before, restrooms are down the back way. There's an emergency exit just around the corner here. Always like to make sure people are aware in case something happens. This is our penultimate event in this academic year. We have one more event coming up. It's actually on May the 7th, or excuse me, Thursday, May the 9th, which is actually after commencement. And when we do those events after the end of the school year, we very often try to find someone who might have a topic that will be as of interest to the local community. So we're going to have Dr. Lily Ferris from the Computer Science Department, who's going to give a presentation on cybersecurity. She's been doing research on phishing scams, and so I'm interested to hear what she has to say. And because we know that after commencement there aren't as many people on campus, that event will be at 6:30 in the evening on Thursday, May the 9th, and we'll actually do that down at the Martin Public Library. And so people from the community can get in there a little bit more easily for that event. But I'm always reminding people to keep an eye on our webpage. We have a Facebook and an Instagram account. We also post a lot of our events on Runway, so people can find them there. But for today's event, this is part of a series we've done during the spring semester looking at women in STEM topics. And so, as we know, there's been challenges in getting more women involved in STEM fields. There's been traditional barriers to that, but I think a lot more encouragement now. And so the events that we've had this spring have been looking at the ways that UT Martin has been involved in encouraging women to be involved in science, technology, engineering, and mathematics. And so today's speaker is a perfect example of that. Dr. Violet Boom is an assistant professor of biochemistry in the UT Martin Department of Chemistry. She completed her doctorate degree actually in Nigeria, and that might be interesting areas to ask questions of her after the event. Her experience has been literally all over the world in different settings. She has her PhD, I wrote it down here, from the University of Calabar in Nigeria. If you go to her resume, her vita, you'll find that she has an incredibly long list of peer-reviewed journal articles that she's produced, conference presentations, and in her various roles, both as a student, a graduate student, and as a postdoctoral researcher, and now as a faculty member, she's been involved in over $1 million worth of externally funded research through the years. So lots of different effort that she's been involved with. I think it's great that she's held teaching positions at the University of Wisconsin-Milwaukee. She's been at Cameroon, and I think most recently in San Diego State. And so a little bit of everything all around the world. She was also awarded a Fulbright Fellowship for her postdoctoral work at Northwestern University. She's really committed to student research, and for those that were involved with our student panel a few months ago, it was great fun to see students that had worked with her on projects, not only helping her with her research, but her helping them with their research, and hopefully we'll hear a little bit about that today as well. But without further ado, I'll turn things over to Dr. Violet Boom. Maybe you can help me welcome her to the stage.
Hello. Yeah, this is working very well. Good evening, fellow colleagues, dear students. It's an honor for me to be here today to, tell you or share with you some of the work that we do in our lab. And as you can see from the title itself, it says we're using light, to kind of bust open microorganisms. That's what it's all about. I've already been introduced, but I'll do a little more introduction of myself. I grew up in Cameroon, which is I don't know if the pointer's working well. Cameroon is in Central Africa, and I did my primary and secondary education in Cameroon, and then moved to my tertiary education in Nigeria, right here. So I was in the University of Calabar. That's where I had my master's and doctorate degree in biochemistry, and I went back to Cameroon to teach. I was very fortunate to be awarded the Fulbright Research Scholarship as a postdoc to go visit Northwestern University. I was there at the Feinberg School of Medicine. I was there for about almost a year and a half, and went back home, because the Fulbright scholarship, you would have to go back and train others. And after 2 years, so my 2 years period, time was up, and I was right back in the States, because I had a nice experience here in the United States. And so I returned to the University of Wisconsin-Milwaukee as a research associate, and then moved to San Diego State University, when I had a grant that was funded with by Careware, and in collaboration with an associate at San Diego State. So I moved to San Diego State, and now I'm here at UT Martin. So that's my journey. Before I go on to the presentation, I would like to just shine light on the students who've done this work. So a lot of the work that I'm going to be talking about today have been done by fantastic students at UTM Martin here. These students are driven, they're very smart, and they put in the work. And so you can see Lauren, I don't know, my pointer, well, I'll go back one more. So Lauren is at the extreme left, and Justin, Lydia, and Deja smiling, taking a selfie. We have a new member in the lab. Javier is not in that photograph, but he's here in the audience. And so we were having fun. That was last semester when we had a poster, I think the STENS Research Forum, with developing our posters for the work that we had done, and we're just having fun. I like to show this picture of this student. Her name is Kine. She worked with me in San Diego State University, and sometimes it's not the quantity, it's the quality. So when Kine came to the lab, she's a medical student. She needed a gap year, and she came to the lab to work with me. It was just 2 of us in the lab for 1 year. So we used to call ourselves the Girl Squad. But in that 1 year, we produced 2 publications, Kine and I. She was so focused and driven. We still have one that's shelved. We're still going to get to it. We haven't gotten to it, but I just wanted to show that, students, it doesn't matter that you have 10 or 5 or 1, it's just the dedication and how driven they are, and you can really achieve a lot. So here's my presentation format. I'm just going to talk to you about the antimicrobial activity of UV, violet, and blue light. I'm going to give you a historical perspective of it and the summary of my early antibacterial findings. We're going to talk a little about the potential mechanisms that might be involved, some virucidal testing that I've done as well, cell viability studies, and some students' presentation, and then we'll wrap it up. I couldn't have gone on without putting this picture on. We're talking about light. And so when we had the eclipse, that just reminded me, just further reminded me of how important the sun is. Without the sun, I don't think life on Earth would be as it is right now. And so this was a reminder, in as much as it was a cool event. And this picture was taken by one of the students, Justin, and he shared it with me, because I didn't go that far. I was right here at Martin. But I think he went somewhere where he got a full shot of it. So that just reminded me of the importance of the kind of work that we're doing in the lab. So before I start, this is not a novel. This is not new. Back in the Egyptian era, you had the gods. So you can see right here there is a goddess, Nefertiti. I don't know if some of you have heard about that goddess, and Akhenaten. They venerated the sun. They have 3 kids. They would go up somewhere that was just designated for them, the king and the queen, and the kids up there to just venerate the sun. To them, the sun was a god. And when they did that, they had this healing, they felt like refreshed and revived. But they believed that it was the God that was doing that. But we know today that it wasn't a God. It's the sunlight. So sun has inherent energy in it that can lead to the production of ATP, we know now, which is very, gives you energy in the system and can drive metabolic processes that help you feel better. So that's why they were feeling better, not for the reasons that they thought they were feeling better for. And then one of the pioneers of light therapy is Niels Finsen. He was a Danish scientist. He won the Nobel Prize in 1903, just a year after he died. He passed. But Finsen, when he used, it's called a Finsen lamp, he used a UV lamp to actually treat tuberculosis, lupus vulgaris, as tuberculosis of the skin and the bones. In that era, surgeons, what they would do is that they would just chop out a big chunk of that tissue and even sometimes the bones. But we know that bacteria, you cannot take away bacteria by just cutting out some tissues and bones. It's going to spread. And so even though they were doing that, those interventions, the children were not cured, they were deformed, crippled oftentimes. And so when Finsen started using his lamp, UV lamp, of course UV would inactivate microorganisms. The UV lamp inactivated mycobacterium, which is what caused the lupus vulgaris. And so that was the birth of light therapy as we know it now. When Finsen passed, you had this Swiss scientist by the name Rollier. So Rollier was the first one to actually really have a clinic and had a solarium. They call them solarium. So he had one on top of the clinic where he would have kids with tuberculosis, go up to the clinic at a certain point in the day, and they would just, soak in, have a sun bath, and they felt much, much better. So that attracted a lot of, people in Europe all over to the Swiss Alps. And so, but why did that stop? Because of the discovery of penicillin. So Alexander Fleming in 1928 discovered penicillin. And penicillin was like the magic drug because when you take it, it would inactivate those microorganisms, including mycobacterium, tuberculosis. It would do that. And so that's how light therapy slowly just waned, just went down. This is some fun pictures of, you can see the solarium that they had those days. You see the kids on the rooftop. And this is Rollier's clinic. So he had two of them. He had one, and then he blew up to the second one because you had a lot of people coming in. And right up here in the Swiss Alps, right in the mountains, you find that they would go right to the rooftop, and these kids were always feeling better. And that started being replicated in the United States as well. So in Virginia, that picture's a little, because I had to fish it out, I had to get it out from 1929. In Virginia, the University of Virginia Medical Center, they did the same thing. They would take the kids all to the rooftop. You can see the beds up there to the rooftop to just get some sunlight. But we're coming full circle. So we're doing light therapy again. Why? Light therapy had waned, but now we're coming full circle. Why? Because now we know that a lot of microorganisms are resistant to what? To the antibiotics that we have. Even before the antibiotic hits the shelf, you already have resistance. And so the microbial community was looking for ways in which, so if you have any alternative therapy, it's welcome because these drugs take a long time to get approved, and when they get approved, they take a shorter time for the microorganism to develop resistance. And so alternative therapies were welcome. And so that's why light therapy started surging again because of the resistance. Now, when we talk of light therapy, it's what we see is the visible spectrum, okay? So it's a range, and you can go from the ultraviolet, near ultraviolet, far ultraviolet, and then you also have photodynamic therapy. Now, photodynamic therapy is if you have, for instance, like methylene blue, and you use light on the methylene blue. That's you're regulating the light by having a pigment that's attached to it. That's photodynamic therapy. That's not just phototherapy. So phototherapy is just having light alone without any dyes. I just wanted to share with you, I know a lot of you have seen this electromagnetic spectrum. So where are we working? What area are we looking at? I circled it right down here. So you can see we're looking at the visible spectrum from 400 nanometers to 700 nanometers. Okay, so if you go the shorter wavelength, it's UV ionizing radiation. If you go the longer wavelengths, you have infrared, microwaves, radio waves. But we're looking at the region between the violet and the blue right there. Now, the red, the visible spectrum, all of that has some application, medical application. The red, we know, is usually used for wound healing or to boost your energy or for anti-inflammation. That's what the red is used for. But the photons in the violet region and the blue light region are strong enough and powerful enough to be able to be antimicrobial. So I was just blowing this up for you to see the region we're working, 400 to 480. So Novothor, which is a company in England, and I know the CEO, they have developed these light beds, red light beds, where people, you know, would go in there, have a treatment session for about 30 minutes and become revived, rejuvenated. Why? Because of, you know, ATP production and a lot of energy. So this energizes people. These beds are not that cheap, but, you know, they do the job. Now, I work with Careware. So Careware has funded us for over $600,000 already. So Careware is another company. They produce flexible substrates. So these are wearable ones. So they're not just a live bed where you have to go inside, but they're flexible, and they would, you know, make them in such a way that they would fit into any part of the body that you wanted to use them for. For instance, you can see one at the ankle, you can see one right there at the vertebral column. And so, and they could, you know, get different wavelengths. So I work with them, and I can, you know, work on bacteria and tell them what wavelengths to use, what specifications they should make, and they'll make it. So there's also lighting panels. You have panels like this that, you know, just like our regular lighting panels. And the goal of this is, if you have, for instance, and this was actually developed when COVID hit, then a lot of sterilization was needed in environmental decontamination, I would say. And so these light fixtures were being produced, and I was, you know, there to test them and find out if they were going to be antimicrobial or not, or antiviral, as the case may be, or not. So before I go on, I'll just define some few terms that I'm going to be using. And so that, and these are terms that you're very conversant with. For instance, power. So if you have a device that you're using, say, what's the power of the device? What's the power of the device? Now, power is the amount of energy that you can transfer per unit time. That's how power is defined. What about irradiance? That amount of energy that you can transfer per unit time, multiply, you're taking into consideration the surface area that power is, the device is occupying. And what about radiant exposure or fluence? It is taking into account the power and the area times the time. So it's like a dose, treatment dose. And so for instance, let's say if I say 7.56 joules, what that would mean is that I have a device that has a power of 0.03 watts or 3 milliwatts, and it's occupying per centimeter, an area of centimeter square, and I'm irradiating for 42 minutes, and it's in seconds. So if I multiply 42 minutes by 60 and times the power, I'll get the dose. So that's the dose. Just like you have milligrams in drugs, in light therapy, you have dose in joules per centimeter square. So we've successfully suppressed a wide range of bacteria with just commonly available diodes. If you had a red laser, like even this, if I had a pointer, this pointer I was working, it's a red pointer. It depends on what intensity it is, you would be able to get some amount of energy from it. So I know this is a little gross, but I'll just talk about this. We know, if you've heard of MRSA before, MRSA has been a serious, serious problem in the US and all over the world for some time, especially in the hospitals. Sometimes it looks like a spider bite or a wound, but it actually looks like that, and it's very purulent, you know. And so we have been able to inactivate methicillin-resistant staph, which is, from the name itself, methicillin-resistant. It's very resistant to antibiotics. And so we were very excited when, you know, we inactivated MRSA. Another bacterium that we're looking at is Propionibacterium acne. You know, that causes acne vulgaris. And we know that in teenagers you have this breakout all the time. And so usually what you have in there, you have P. acne, and then of course the inflammation sets in. You have all those comedones that you can see. So in the early back in, that was like 5, 6 years ago, we're using a handheld device. So this is another company that funded us, Dynatronics. So Dynatronics had this handheld device that had some diodes in it, about 36. So let me test your knowledge of irradiance right now. So if we have 5 centimeter square applicator, an average power of 150 milliwatts, the radiance therefore would be what? 30 milliwatts per. Why? Because we're just dividing that, remember, by the area. So 150 divided by that is 30 milliwatts per centimeter square. Yes, maybe. So if you look at the first, I don't know, microbiologists will relate to this better. That's how you have bacteria in dishes. So you can actually see them forming colonies. And so if you look at that's MRSA, that's methicillin-resistant Staph aureus that we cultured. You can see a control plate. So we actually would grow it, and we put the same amount in all 4 dishes. Okay, and then we irradiate it with doses of 55 joules per centimeter square, 110, and sometimes we even go higher to make sure that everything is done 220. So if you look at it just pictorially, you can see that the 55 inactivated some but not all, but the 110 and 220 joules, kind of inactivated all the MRSA. So that was very exciting, because if we have a protocol that can do that, and MRSA is a problem in the hospitals and in the environment, then that would be a tool that we could develop to inactivate MRSA. We've done this with salmonella as well. So there was a group that wanted us to test against salmonella because they had birds. And these birds were being infected by Salmonella enterica Heidelberg. So we have typhimurium, which is what is typhoid fever. Typhimurium causes typhoid fever in humans, but you have Heidelberg in animals and birds. And so we did the same protocol. We used our 55 joules per centimeter square and irradiated it, and then you have 110, 165, and 220. You can see when you go up to 165 and 220, you'll be able to eliminate a lot of this. And so the group that wanted us to do it was very excited. We got data like that. And this is just, representing that in graphical form. It's the same thing that we see. I like seeing the pictures. It makes more impact than the graphs. So now what we're working with now, Careware has this wearable substrates. And this wearable substrate, we have now moved from continuous. So what I showed you was continuous wave. Now we've moved to pulsing the light. So if you pulse the light, it's called duty cycle, 33% duty cycle, which means that the light is on 33% of the time and off 67% of the time. You might not even see it because it's microseconds. And so this patch that you have here, this is magenta. So it's mixed. You have some red, you have some blue. And this is for acne, because when we're working with acne, we want to inactivate the bacteria, but we want to stop inflammation as well. And so that would help. And this patch has a wireless controller. So this is a finished product. But in the lab, we have prototypes that we're using. But this is a finished product. And this has been patented. So we have this patented already with the company and has been FDA approved by the company. And I was very instrumental in the FDA approval. I want to give myself credit for that. So they're marketing it. It's in the market, and they're using it. So these are prototypes that we have in the lab that we use. And you can see they're flexible. You can adapt them in whichever way you want. And that is for the arm, and this is for the forehead. So this is magenta, and this is for acne, if you want to do acne. And that's my arm right there, anyways. Let's use myself as a guinea pig. So the setup is pretty simple. For acne, because it's anaerobic, we have a chamber, and then we put a sachet inside, and that sachet just sucks up all the oxygen, because P. acne likes anaerobic conditions. It will just suck up all the oxygen, and you can have your substrate. You see dishes under there. You have your bacteria in the dishes, have your substrate on top. I just irradiate them. And sometimes it depends on the protocol. We irradiate for 30 minutes or 42 minutes. But the protocol we're using right now, we're irradiating for 42 minutes, give you some downtime, irradiate for 42 minutes, give you some downtime, irradiate for 42 minutes. So 3 times a day, just like you're taking your medication, same thing, 3 times a day. And then we incubate them and then come back the next day to look at the colonies. In acne, you have to come back after 3 days because it's slow growing, takes a long time to grow for you to see colonies. So this is just a typical example. Now remember, the fluences that I showed you before were very high. Now we're down to 3.6 joules, which is really remarkable 3 joules, 3.6 joules, and 5 joules. That's very, very low energy, sometimes even lower than the white bulb. So this is not too much power that would destroy cells, as we will see. Now, if you look at acne, you can see the control right here, 3.6 and 5 joules. You come back in 3 days, you don't find anything on the plate, it's all gone. So we know that this protocol inactivates acne. One of the problems in microbiology, if you're a microbiologist, and in medicine, of course, is you have biofilms. These bacteria will form biofilms. And when they form biofilms, the antibiotics that are being used are difficult, very difficult to penetrate into the biofilms, because this is bacteria talking to themselves, cross-talking to themselves. And they form like this canopy that protects them. And so you have to kind of use to kind of scatter that colony, that biofilm, to be able to get access to individual bacterium in it. And so we use our technologies. If you look at up here, we stain them. So the staining will help you see the bacteria. So the green is alive, and the red staining is dead cells. And so this column here is supposed to be just a normal bacteria colony. But even in the normal colony, you have some of them are dead. So you have few dead. So when we use our technology, you could see how the thing had blast open the biofilm. We didn't, inactivate everything, but we're very excited because you could, open the biofilm, and then you can do multiple treatments to be able to clear everything. And you can see the amount of dead cells is a lot. So what are the mechanisms? We now know that this protocol inactivates microorganisms, but what are the mechanisms involved? There are a couple of them, but I'm going to talk about 3 of them today. One is the release of reactive oxygen species by excited photosensitizers. We'll talk about that. 2, we've seen that it alters the membrane potential of these microorganisms. And if you know about membrane potential, if you disrupt the membrane potential of the cell is gone. And also, 3, there is impairment of cell division and replication. We use transmission electron microscope to see that. So I'll just explain this just a little, go through the mechanism. So production of reactive oxygen species, how does that happen? So you have light 400 to 480 nanometers, hit the microorganism. The microorganism has a photosensitizer or a pigment in it, and that pigment gets excited. So the energy that is in light is a form of photons, excites that pigment, and the pigment gets to an excited state. But it doesn't stay at excited state, it has to drop down to ground level. When it drops down to ground level, there are 2 things it would do. 1, it would emit that energy in the form of fluorescence. You will see light. 2, it will get to the triplet state. When it gets to the triplet state, it would now have that energy that interacts with things like molecular oxygen in type 2, or other substrates in the system in type 1. So for instance, you have the lipid bilayer. The bilayer is made up of lipids. If you have those ROS, reactive oxygen species, hitting the bilayer, it's going to cause lipid peroxidation. If you have peroxidation of the bilayer, the membrane integrity of the membrane is gone. And when it's weak, there's osmosis, water gushes into the cell, the bacteria is gone. So, but then before we did that, we want to say, what are the pigments? So if we're saying that there are pigments in bacteria, what are the pigments that are present in this bacteria? And so we looked at methicillin-resistant staph, that's MRSA. So if you do a fluorescence, spectroscopy, which means that we would shine 405 or 410 or 425 or 450 nanometer light on the bacteria, and then we would measure what it emits, the light that it emits. So usually it emits in the red region, and you can see that. So when we looked at the emission spectrum, we realized that it has porphyrins. MRSA has porphyrins, and porphyrins absorb very well 405 and 470. So that's likely what is happening. They're absorbing the energy from the light and producing reactive oxygen species. And so here we're looking at the membrane potential. So the membrane usually, the membrane has positives and negative sides. So inside the membrane you have potassium ions and some anions. Outside the membrane you have sodium and chloride, right? So if that's how it stays, it's neutral. But if for some reason the potassium ion leaks down the membrane, then you have a potential, because it's a difference. It's more negative inside and more positive outside. So you have a potential. And this potential, usually you have pumps that would push back the potassium inside. But if that membrane potential is disrupted in such a way that the membrane integrity is affected, then you can have things going in and out of the cell. The cell usually is semi-permeable, doesn't let things go in and out. But if that happens, you can have things going in and out. If you see right here, so we stain the MRSA, so you can follow individual bacterium right there in a certain field. And so if you stain them, this red indicates that they are alive, okay? And then we irradiate them with our protocol. Almost immediately, you start seeing the same, and you can follow them, 1, 2, 3, 4, you can follow it. It goes green. You see how green it is. So it's dead. The membrane potential is compromised, and you can start seeing them staining, dying as they're going on. So we knew that the energy in the light photons affected the membrane potential.
Can I ask a quick question?
Yes.
Knowing nothing about it.
Yes.
So does it stay that way, or does it have to continue to be irradiated?
No, so we irradiate, so the protocol is 30 minutes. We irradiate for 30 minutes, and we're done. Take it out. And then you just continue monitoring fluorescence, and you see that you're having that red, just saying they're dying. So here is, we wanted to see the ultrastructure of the bacteria. So we're using transmission electron microscope. If you look at that on this side, these two here are, you have MRSA that is dividing. You can actually see the septum, right? You see it dividing. Healthy cells, they're fine, they're growing very well. When we radiate them 42 minutes, you can see they radiated the treatment group. You don't see them easily dividing. This is trying to divide, but you can tell that the wall is already perforated right there, and it's having a problem. If you look at the nucleus, so here you have 2 nuclei. The nucleus divides, you have 2 nuclei. But if you look at it here, it's kind of compact. It tells you that it's not even willing to. So there's something going on right there. You can tell that the cell is not willing, it's not dividing as it should. And this is even clearer. If you look at this, you can see this is really healthy. And it starts by you having the nucleus that's spreading, try to divide. And you can see clear septum right here. But here you can see, look at that. So you find out that once you irradiate this bacteria, it affects the cell membrane or the wall of the bacteria. We know that now because we've looked at it. These are like thin slices. Can you imagine like using a gold knife to slice a bacteria that you don't even see? Okay. And so just like medications, we now went into what is a dose? What regimen? How do we get a regimen? You know, when you take an antibiotics, it's going to say 500 milligrams a day or 1,000 milligrams a day. So for light, are you just going to shine the light on the microorganisms? You got to be able to get some dose regimen. And the way we were able to do that was developing this device. So this is a spectrophotometer right down here. So in here we have like our dish with the bacteria in it. We have our light source that's up there to irradiate it. And we have a filter after the light source. So what the filter does is the filter lets in only 4 or 5 go through. If you want only 4 or 5, it's only 4 or 5 that goes through. And then the bacteria is right there, but when we're collecting that, we have a second filter. The filter takes out 4 or 5 because we don't want that going through. Now it's just the fluorescence, which is around 600 and 660 and so forth. So we can collect fluorescence data from there, and that's what we did. Now, we did that on P. acne, and all of these have been published. We did that on, as a publication right there. So we did that on Propionibacterium acne and followed it over time. So, and this work is like 3 months or 4 months or 6 months. So what you see as a final finished product, but it took us months to be able to nail it down. And so here you realize that once you irradiate, you put in a dish in that device that I showed you. Once you irradiate it, we found out that the best irradiation times were, if we do, this is 10 minutes, 20 minutes, 30 minutes, 40 minutes, and you see the fluorescence going down. And then we let it rest for about 30 minutes, it builds up. And then if you irradiate again, you have 40 minutes, it goes down, 30 minutes, 40 minutes, it goes down. So we did that on day 1, day 2, and day 3. By day 3, you didn't see it going. So these are just like artifacts. These are outliers right there. But day 3, it was pretty much flat line, which told us that you don't have, because we correlated the fluorescence to the amount of bacteria that you have, which told us that you probably don't have much left. And that's reflected in these plates. So these are the same plates that we worked with. So you see a control, look at the treatment group. At the end of the 3 days, there's nothing on the plate, and we didn't get any signal. So we can use that for different bacteria. So this is P. acne. It's slow growing, and so we did after 3 hours. We've done that with MRSA. MRSA is really quick. In 30 minutes, you're going to double. So we do that faster. So we could use this for different microorganisms and actually say what our treatment protocol is going to look like. It takes some time, but we can nail it down. And so when COVID hit, at that time, it was like, okay, let's try this on COVID, because the vaccine was not there yet. There was no protocol. People were taking turmeric, curcumin, different sorts of things. People were taking things just to, inactivate the virus. And so we said, okay, we have a tool here which is antimicrobial. We could as well test this on the virus, right? So we had our virus, but remember, we didn't get SARS-CoV-2 because at that time, nobody even wanted to go. SARS-CoV-2 is what caused the pandemic. So nobody wanted to go close to SARS-CoV-2. So we look for a family coronavirus in the same, it's a beta coronavirus. So we look for a beta coronavirus in the same family. So it's just like you have your sibling, right? Something's going to affect you genetically, it's probably going to affect your sibling somehow. So we look for other coronaviruses in that family. And the ones that we looked is called HCOV-OC43, which is a beta coronavirus, and there's HCOV-229E, those two up there, which is an alpha coronavirus. So what we did was we used our irradiation protocol, irradiated it. And then, because they're RNA viruses, we extract the RNA, we quantify the RNA just to see what's happening. That's what we did. And so if you look at here with our protocol, you can see 32 joules, 64 joules, and 130 joules at an irradiance of 12 milliwatts. You can see that we can actually, we had RNA concentration dropped by 50%. You can see that. As you increase the dose, the RNA concentration was dropping. And if RNA concentration is dropping, you can relate that to the virus. So you don't have as much of that virus in that dish as you would think. Now, but then you have to confirm it, because that's not only the thing, because you can have RNA, but it could still be infective. So you have to do a qPCR. So you have to be able to, just like the normal testing, remember when you go to test for, your virus, the virus in your, if you have the virus in your system, or that's what they do. You go in there, they do a qPCR, and they look at the CQ cycle. So the CQ simply tells you when this is amplified, because it's an RNA virus. For those in sciences, my students, there's reverse transcriptase. Reverse transcribes it to what? To DNA. You can amplify DNA and see how much you have in there. And so if you do that, you can see CQ values of non-irradiated controls right here. It's like 15. So at 15 cycles, which means that it's above the threshold. So you have a threshold, but then you have cycle that you start seeing the virus. So at 15 cycles, you see. But with our treatment protocol, you saw them a little later. So it's telling you that it's slow to build up. So when it's amplifying, it takes a much longer time to amplify, which means you have less starting material. And so you can see that how it goes all the way to 20. And that's been published. So because we're thinking we want to have optimal inactivation. We know it's 50%, we're saying. What can we do to improve on this? It's like, okay, people are taking turmeric. So, okay, we're going to test turmeric, which is curcumin. So we had a dish where we had turmeric in the media that had the virus, and then we irradiated that. Or saliva, because some people was like, oh, saliva inactivates this virus. Or just saliva. Does saliva inactivate the virus? We had saliva, and then we had saliva and the light. So we had a combination of both. I'm not going to, this is a very busy table, but I'm going to let you focus on this column alone. So if you look at HCOV-43, which is the same family as SARS-CoV-2, you find out that when you irradiate them, you get up to like, this is 67% inactivation of the RNA. And if you have saliva, it really does a good job. And if you have saliva and the light, it goes down. In curcumin, even better. And if you do curcumin and saliva, you're up to like 10%. So you're inactivating 90%. So you have 10% of the RNA left, which is really, we were very excited. 10%, that's, if you know RNA, sometimes difficult to degrade. So 10% of RNA left, that's what we had, which was very, very impressive. Now, to confirm that with a qPCR, you can see right down here, look at the cycle. It goes all the way to 50 cycles. And usually when you go to do your test, when you get to 30 cycles, That's when they say it's below detectable units. So if you go and do your test, as scientists, we can't just say it's negative. No, it's like below detectable because you leave that little room for probabilities of things. So anything beyond 30 cycle is saying that it's below detectable units. And we could see that all the way from here right down. So supplementing this with actually inactivates coronaviruses. And so we're getting towards the end. One of the last things, not last, second to the last, that we're going to talk about is cell viability, because if this protocol is going to be used on the skin, or even because when coronavirus hit, the company was going to produce nasal buds. So you're going to have nasal buds with blue light into it, with the hope that it will inactivate the virus before it gets to its receptors in the lungs. So, and if you can decrease that, so even with what we had, if you can decrease that, then you give the body a fighting chance. The immune system can fight. Because the thing is, the immune system is overwhelmed, it's not seen this before, and so if we can even reduce that, we give the immune system a chance to fight. And that also brings me to the point that if you have this RNA, even though we had 10%, we didn't see any amplification. So it could just be a carcass, just like, let's say you have a dog, right? And the dog is, dead, not living. You still have the dog, but it's inactive. It can't bite, it can't do anything. So that's what it's like a carcass. So you can have RNA, but the RNA is not amplifiable. And so that makes sense. So we saw that our RNA, even though we had a little left, we couldn't amplify it. And so with human cells, we just because we wanted to test, on the skin you have fibroblasts, you have keratinocytes, and all of that. So we tested this. I'm so sorry you can't see this. It's been published, but I'm going to go to the data right there so you can see that. So if you look at this, these are fibroblasts that are grown in the lab. And this is like a control, and then 3 joules, 55 joules, and 110 joules. Because we want to make sure that if we're going to use this protocol, it should not be inactivating cells. Because those cells are fighter cells. They help, they should work synergistically with what you're having rather than destroying it. And so we saw that. So we used 3 joules. Now we're under 3 joules. We're like 5 joules. So with the 3 and 55, it was okay. But when we got to, if you look at this picture down here, when we got to like the 110, you see that they were still viable. When we checked for viability, the cells were still alive, but they were rounding up. Fibroblasts have like this tendency to spread. They have cytoplasmic spreading. You can see them spreading here when they're happy. When they're not happy, they fold in. It's just like you're in a cold, you want to fold in. So they fold in. So we knew that you get into those doses, they're not going to be that happy, but they're still viable. And these are just measuring things that they secrete. So another thing is that, okay, if they're not happy, if they secrete these growth factors, are they secreting at measurable amounts to control? And we saw that it was, it was a little low, but it was not significant. So here, this is one of the models that we use for wound healing. So if you're doing wound healing, you don't necessarily have to make a wound on the animal. You could do wound healing in a petri dish. Okay, so what we do is that we get fibroblasts and we plate them. When you plate them, you get like a pipette tip and just make a wound in the middle and see how the fibroblasts will close up. So this is an in vitro model. You could test this before you go into in vivo models to make sure that you have something that causes the fibroblasts to move towards to close that gap that you have. And so if you look at this is a control, this is 3 joules, this is 5. You can see that 5 joules even closed faster than the control, which told us that it even had some active. And the cells look very fine and very healthy. And so we knew that if you had a wound and you had to irradiate, you'll be able to have fibroblasts, which is what you need to be able to repopulate that area and get your wound healed. So this is just staining, so you can see it on the fluorescence microscope. Now, one of the beauty of this is that we've been working with these things here. I went to Africa. I had a scholarship to go to Africa last summer that was funded by the Carnegie Foundation. And I went back to my university in Calabar after how many years that I had, 17 years or so, I have not been there. So when I went there, I went to the teaching hospital, and I took a protocol with me to the teaching hospital. And we went there, they were very excited. They said, we have this, we have field isolates. I said, oh, wow, this is great. So these are people who are really sick, and you can actually swap and get the right straight from the, from the wound. And so we got Staph aureus. If you see, look at the control here, we cultured it and we treated it. You can see that our plates were empty at 4, 5, 410, 425, 450, maybe not. The reason why, because it's a little further away from UV. We know that UV would the closer you are to UV, the energy is higher in the photons. But we're very excited to see that we could inactivate field isolates, which means that our protocol can really be translated, you know, to not just on the bench, but can go to clinical settings. So here at UT Martin, I was just going to show some students' posters that we have, my students. So this is Lauren, who's the first author of this one, and this Lauren's right here. So this is the virucidal effect of pulse blue light on human coronavirus. So when I came here, I was like, we got to optimize this protocol and make sure, because sometimes you move an environment, you want to go to a different place, you want to make sure that you can establish the same thing that you saw. And they worked on this with Lydia and Deja, Justin. They worked on this, and we could replicate the data that I had. They could replicate it, and this was used in the SENSE thing, poster. And there's another one. Deja was the lead author in this one, but this is on RSV. You know, was it last semester that was RSV was a big thing in the news, respiratory syncytial virus. And so we said, oh, okay, let's test this on RSV. And we did that. We saw that RSV was inactivated. They looked at the RNA concentrations, and you can see inactivation of RSV. So here, we're hoping to go to London. So the students have worked so hard, and we put in some abstracts. In this case, is the effect of pulse blue light on commensal microflora. You know that the body has its normal commensals, right? And so if you're inactivating this with light, we're checking, does it inactivate other non-pathogenic bacteria as well as the pathogens, or does it not? And so they worked on that, but what we see is that, which we're going to present in abstracts, has been accepted. One of them has been accepted. We're waiting for this other ones to be accepted. Hopefully they will be accepted. We saw that inactivation of bacteria was dependent on the species. So if it was a staph, you're going to inactivate staph. It doesn't matter if it's MRSA or is, for instance, Staphylococcus lugdunensis, which is a normal commensal, it would inactivate it. So it didn't matter if it was a normal flora or not. But then, again, does it really matter? I don't think so, because when you have a wound, it means that the pathogens have already combated the non-pathogens. And so all you want to do, go in there, clean that, and let the non-pathogens repopulate the area. That's what you want to do. And so the students are going to present this. And so the next one, you have the second one and the third one. So we have 3 of those. This is the World Association for Light Therapy, now called World Association for Photobiomodulation Therapy. But we're very excited that we have 3 papers, and hopefully we can go to London in August. And so I'll just wrap up with, like, what are the future things we want to do with students? Like, you know, I mean, I am all for training students, and the group of students that I met here at UTM, they're students that are very smart, and we would like to really work on certain things. So bacteria testing, we're going to continue, and I'm part of an NSF task force that is paying attention to antimicrobial resistance. So I know that is something that is hot, so we're going to continue. And then I'm intending to work with faculty, maybe to produce, chemicals that we can use to inactivate microorganisms, chemicals in combination with light. For instance, I work with someone who actually produced germanium-doped hydroxyapatite, and we, saw that it was antimicrobial. And then wound healing studies, and then maybe we'll get into some cancer targeting. We're thinking the next thing is using the red light, move slowly into using red light and photosensitizer to be able to target certain. So we're going to kind of have that linked to target an antigen using an immunoglobulin, and target an antigen, and we can use a laser to go in. And so that's what we're thinking of doing moving forward. And so just to conclude, I would say that these protocols that we're doing, we're trying to develop light-based technology that can be used to inactivate bacteria in the environment and also in clinical settings. It has several applications. Even like, for instance, if you talk of applications in ambulances, like ambulances, these ambulances, they don't have enough downtime to, wait and sterilize before go pick up another person. So if you could have a UV, not UV, a blue light device in there, UV because it's deleterious, it would cause breaking of the DNA. We don't want that. So, but if you have a blue light in there, then why they drop people, we were contacted by an ambulance company. That's how I know that we could use that. They contacted us like, so if they could drop, you could drop people and then just eliminate the place while going to pick up another person. That will help to sterilize the environment before another patient picks up something. So it has tons of applications. And I was just going to show you, this is the CEO of the company, Careware, that produces those patches that I work with. It's me there. And we have a pilot study right now in PACNE in San Diego, and those people are working. And this one is my mentor. He's one of the founders of PBM, and he's my mentor. He taught me a lot of things that I know right now. He's almost retiring, so. And well, even on graduation day, I was just going to show this picture. This student came in even on graduation day, and that was a lab. And students could be formed right here. So this is us right here. So now this is UT Martin, because some of the work I showed you was done in San Diego, and some of the work was done at UT Martin. So that's Deja, this is Justin, and this is poster time. So we had, you know, KLS and poster time with the students. Justin, you guys were not there for posters time, so that's why your pictures are not here, because you were not there during this poster. And that's us. So I've just highlighted the students that I've worked with here at UT Martin. You can see it. They're great students, fantastic students. So I'd like to end by thanking the Department of Chemistry and Physics, and also the Department of Biology, because we do a lot of work in the biology department, and they've been very gracious to us and give us their space to work. I always like ending with this quote by Einstein. The most incomprehensible thing about the world is that it is all comprehensible. So that's my ending. Thank you.
Time for a couple of questions.
So if someone's dealing with acne, they have a device that you can put on for 30 minutes?
Yes, yes.
Can you do the same by just going and laying in the sun for 30 minutes?
Thank you. That's a very good question.
Repeat the question for those.
Yes, let me repeat the question. So the question is, if you have this protocol that we're using for acne, right? Let's say 30 minutes, you're ready for 30 minutes. Can someone just go out in the sunlight? Because sunlight, you know, can you just go out there and just, you know, get yourself irradiated and it works the same thing? Yes, sunlight inactivates, but the amount that gets to you is not enough. It's not adequate. Because we know, like, UV gets to us, but the ozone layer traps UV. So what we get down is blue. We get a lot of blue light coming down, not too much. UV a little. That's why they always say, oh, don't go out when you have a UV index of so and so, don't go out. It could be dangerous. But you have a lot of blue, which is longer, getting to the earth surface. So yes, it will work, but the dose is not going to be effective. So this is in the lab where you kind of synthesize. You get the power in that you need, but you can still get some of it, but it's very, very, very minimal. Yeah.
So I have a question, and I have a microphone too.
Yeah.
My question is, I can imagine those devices that you showed being really useful on acne or a wound. What might be the application to actually treat someone who has COVID? I mean, is there a way that you'd be able to treat an entire body? Because
Yes.
I have to imagine that the infection is everywhere.
Very good question. That question is a very excellent question, because we know that this is systemic. So when you have a systemic infection, it's in the blood and all of that. Now, UV light and blue light, they're not that long. So red light can penetrate the system, can go. Red light is very long. Red light does, but red light is not that antimicrobial. Red light is more for energy, for you know, building up of things, and blue photons are very strong. So no, you cannot use it for systemics in that regard, because on the surface, yes, because blue light penetrates like about I think 1 millimeters. That's how much blue light can penetrate. But red light can go up to like 4, 5 millimeters, so it could go really deep. But the company that was going to use it for COVID wanted to produce some boards, like, you know, nasal boards that you could put in your nose, and then it irradiates up right there. And if they did that, their hope was that they were going to inactivate some of them before, because we know that COVID is going to go look for the lung cells. It has receptors too and attach to them. And once it binds, but their hope was to get it while it's still in the process of going in. And also, it could be used for like catheters. So people have catheters. You can have catheters that are inserted into the system. And we know that people who have catheters have this point of locus where you have bacteria infection. That is always the case. So now what they're doing, they're trying to have catheters that are lined with blue light. So that locus, so once you have your catheter, it's going to be irradiating that area and prevent it from having, you know, a bacteria stronghold. So there are certain things that can be done, but there's a limitation as well. And that's where, if you have medications, it could be synergistic with that. Because we also know that some of these medications, especially for topical things, don't get to the skin very well because they are systemic. So if you have things that affect you on the skin, you're not going to have too much of your medication getting up to the skin. So if you have something like that, so it's usually something that is a tool that could be complementary to what you already have.
Great. One more question for anyone?
I have a question. The skin cancer, you said is future study for possibly using red light?
Yeah, so red light is, well, not skin, cancer inside, not skin, yeah. Yeah, so for in the surface of the skin, because cancerous cells is like cells that are not responding to, you know, there's like, they've gone haywire, you know, and they replicate and things like that. Now they have markers. So what we're trying to do is that those cells have markers. So there's certain antigens that will differentiate them from normal cells. And if you can develop antibodies to those markers, so you can actually tag a dye to the antibody, drive them to where the cell, and it's not gonna, so it's not gonna, it's just like chemotherapy, it's not gonna kill indiscriminately. It's gonna go all the way to where it finds its antigen and hook onto it. And then you can come in with your laser, if it's internally, going with a laser and get that cell out.
Great. Well, I want to thank you very much, Dr. Brumah, for being with us today. We have been doing a live stream of this event, which is also a recording. So if you're interested in this, or if you talk to people who weren't able to attend, I know we had some listeners online. I was checking while we were here. That replay will be ready probably within a day, and you can go to that same link that was in the announcement for this event and replay this in its entirety or portions that you want. But I hope that the folks that here might help me in thanking Dr. Brumah for an excellent presentation. Thank you.